Hello and welcome to GenQA's Focus On webinar, which today is discussing selecting tools and assessing and classifying variants. We've got a fantastic panel with us today who are live and are going to be able to answer your questions at the end of the presentation, which is fantastic, so thank you. I am Sandy Deans, I'm Director of GenQA, and I'm also joined today by three of my colleagues who all work in the area of rare and inherited disorders and delivering the external quality assessments for molecular genetics. So we have Dave, do you want to say hi?
Hi, afternoon everyone.
Which EQAs do you cover, Dave?
Oh, I do a whole variety of the rare disease, some of the technical EQAs like NGS, newborn screening, various.
Great, thank you. Farah?
Hi, I also work with Dave and cover lots of molecular genetic EQAs, the NGS EQAs and PGT EQAs.
Thank you. And Becky?
Hello, I cover some of the molecular genetics EQAs as well as pharmacogenomics.
Great, thank you very much, everyone. So we're also joined today, as I say, by our expert panel. We have Dr. Julia Baptista, who's a clinical scientist and currently the scientific training officer at Sanofi's in London, in the UK. And we're also delighted to be joined today by Dr. Swati Tomar, who's the product manager for the Rare and Inherited Diseases section in Sophia Genetics. And Swati is also going to, as part of our presentation, introduce Rolf Funt, who is going to talk about the clinical use and the actual using the tools that as we move through the presentation. So if we move on to the next slide, please. We're going to start with Julia, who is going to talk about the tools for assessing genomic variants in rare disease. And then Swati is going to talk about enhancing variant interpretation, so about leading you to answer from a vast universe of data, which really we're all struggling, and it's quite challenging at times to be able to work your way through the data that's available and come with a clinical interpretation. And then, as I say, we'll have an opportunity to answer your questions live. So I do encourage you to put your questions into the questions box that hopefully you can see on your screen, and we'll work our way through them after the slide presentations. So if we move on now, so it's my great pleasure, as I say, to introduce Julia Baptista. So Julia has been a clinical scientist in the NHS for many years and also an educator with over 25 years experience in genomics. As well as her current role, she's a visiting senior lecturer at King's College London, and she also is a board member of the European Society of Human Genetics. So Julia, greatly appreciate you coming today, and you obviously have a vast experience and knowledge of using the tools for trying to interpret the genomic variants for rare disease. So over to you, please. Thank you.
Thank you very much, Sandy, and thank you for the invitation and for that kind introduction. Please, can I have the next slide? So what I'll try and do in this very short presentation is to cover some key concepts of variant interpretation in clinical genomics. There are so many tools that are available out there for us to interpret the variants, but I'm going to focus very briefly on GNOMAD, ClinVar, and Decipher. Please, may I have the next slide? Thank you. So why do we need these tools? So as Sandy alluded to, we are pretty much all doing next generation sequencing technologies, from large panels to exomes and genomes. We sometimes are overwhelmed by huge amounts of genomic data. And whilst we have learned about some tools that can enable us to be a bit more efficient on how we look at variants, we are nonetheless left with some variants that require us to do a proper classification. And most importantly, we need tools to lead us to the right answer for our patients. Next slide, please. So pretty much like building a picture, we clinical scientists are looking for different pieces of evidence that we collect, understand, and put together. We are asking biological questions to the data and to those data sets, and we want to reach an answer, a final answer for a variant. So that could be, is the variant benign or likely benign, and therefore I don't need to include it in the report? Is it likely pathogenic and pathogenic? Or does it explain the reason why I tested the patient in the first place? Or I might be a little bit in a region of uncertainty and end up with a variant that has uncertain significance. And I might or might not then need to do additional work to be able to interpret that variant. Next slide, please. So a lot of what we do, I'm not going to go too much into the ACMG guidelines, but the purpose of this slide here is to really give a framework of what we do, why we do it the way we do. So most of us are using the ACMG guidelines or a variation of the ACMG guidelines to guide us on the questions that we need to ask to the different tools to get the answers for the data that we are interrogating. Now, what I wanted to say as well is that the original paper from Richards and colleagues has now been published in 2015. So from then until now, we have gathered a huge amount of knowledge and experience and understanding, not only of the guidelines per se, but also of genes and conditions and variants. So it's really important that we move on with the times, and CleanGen has been absolutely instrumental in providing us with knowledge to make sure that we are using criteria that are up to date and refined. So CleanGen aims to support not only the implementation of the guidelines, but most importantly, the refinement of the guidelines to make sure that we are using the tools appropriately and interpreting biological information in a manner that is updated. And in that way, we will have harmonization of the conclusions that we reach from the data. And that, of course, will improve consistency for patients. May I have the next slide, please? So what are those biological questions that we are asking to the data and to the tools that we are using that are being framed by the ACMG guidelines? What is the predicted effect of the variant that I'm seeing in the protein? What phenotypes does this gene cause? And you might notice that I used phenotypes rather than phenotype, because more and more we are seeing single genes causing multiple conditions. And some of those conditions are novel, recently discovered, so we need those updated tools and knowledge to interpret the data. Is my variant in a key region of the protein? What is the context? Do I know the disease mechanism? So those are some of the questions that we ask when using the data. So if we move to the next slide, please. So what you see here is, if you like, a categorization of some of the data that I mentioned and of those questions. So if you look at this diagram, and I start at 12 o'clock in the diagram, you will see the literature bubble there. So I want to interrogate the literature, and as a tool, I might use PubMed, I might have access to Mastermind or the Human Gene Mutation Database, for example. If I have a variant that is a splicing variant, I might want to use Splice AI to interrogate that variant. Even missense variants can be splicing variants, so I might want to use that tool for missense variants as well. And typically, most laboratories in the UK have now moved to using Revel as the tool of choice to interpret the impact of a missense variant on the protein. I might want to collect information on functional studies. Are there any? And might need to do a deep dive into the literature to find the answer to that question. And next on that diagram, I'll move to population data. And this is where most of us typically will be using GNOMAD as the tool of excellence to ask the question, has this variant been seen in population data before or not? So if I now move to the next slide, please. Just to introduce GNOMAD, probably many of you use GNOMAD, and I just wanted to touch on some of the power that you will find in version 4 of GNOMAD. So we now have huge amounts of genome and exome data in the database, but some of those premises that apply to version 2 and version 3 also apply to version 4, and that is that although most of the data comes from individuals that are free of severe early onset pediatric disorders, you need to be cautious because now there's a huge amount of biobank data where you can have individuals that have severe conditions, but they are in the mild spectrum, okay? So you need to consider penetrance and reduced penetrance in particular, and other biological mechanisms like mosaicism and acquired variants. All of those biological premises still apply to version 4 data, and much more so. And the other thing I wanted to say about version 4 is that because of the huge amount of data that is now there, we scientists that work in rare disease are now seeing variants that are rare, that cause disease, but are in quite high numbers in GNOMAD version 4. And we need to kind of reframe the way we are used to looking at data in rare disease. Can I have the next slide, please? Thank you. So this is an example, an extreme example of a variant that you can see in GNOMAD that has over 200 heterozygotes in GNOMAD, but is still a pathogenic variant. I'll explain why this is pathogenic, but what I wanted to do here as well is to touch on how much data is in GNOMAD. So we have to think of GNOMAD as data set that is much more than allele frequency. So for example, you can get in silico predictors information from GNOMAD. You can get links to ClinVar as well. So it can be a really nice resource to get you started when you are assessing your variants and interpreting them. So this variant specifically is so frequent in GNOMAD, if you like. And the biological reason for that is there in GNOMAD. If you explore the page and look at age distribution, genotype quality metrics, you will see the data indicating when you have acquired variants. So in this specific case, what I'm showing there is that the allele balance for this variant is skewed from 50%. So most individuals that are in GNOMAD and have this variant don't have it at 50-50. So that is an indication of somatic mosaicism. So GNOMAD has that richness of data. Can I have the next slide, please? Another extreme example of a variant that is common and is not benign. So actually this variant in versions 2 and 3 of GNOMAD surpassed the 5% frequency that would automatically, if you were applying the guidelines from 2015, you would automatically discard this variant as too common to cause rare disease. But in version 4, the frequency is now lower than 5%, and it stands at around 3.8% for this variant. And it's really just to show you, the kinds of numbers that you can now see in version 4. And the reason why this variant is so common is because it's low penetrance. So you have individuals that have the variant but don't have the phenotype. And the ClinGen group has a small list of variants that are common and are not benign. And you can access that information on the ClinGen group as well. Can I have the next slide, please? So I want to move on from GNOMAD and touch on ClinVar and touch on some of the features that make ClinVar such a great tool for assessing variants and collecting information. So typically here we are asking the question, has anybody seen this variant before? And is there any evidence that the variant is pathogenic or benign from the submissions that are in ClinVar? As you know, there are submissions that are archived into ClinVar from different sources, so it could be from research groups, but the majority is from clinical laboratories. And the really exciting development that is happening in ClinVar lately is this revision by expert panels, again, driven by ClinGen and their working groups. They are now labeling these variants with a 3-star rating and a clear label there where you can read reviewed by an expert panel. This is extremely powerful for laboratories because it allows us to then not have to do an independent assessment of those variants because the ClinVar, the ClinGen group has done that. Can I have the next slide, please? So still staying with the ClinVar, an example on the other spectrum of a variant that only has a 1-star rating. But you can see that there's a lot of evidence from the different labs that have submitted their conclusions. Nonetheless, those conclusions are conflicting, and when you're trying to evaluate that, you shouldn't be trying to choose one or the other. You really have to, unfortunately, go on and do an independent assessment of those variants. Nonetheless, it's very useful to see what other labs have seen, and you can also contact the submitters and discuss some of that evidence. So ClinVar is extremely helpful. Next slide, please. Now I'll move on to Decipher, and in the UK, many of us use Decipher as a one-stop shop for all things variant. Most laboratories use Alamut and other decision support tools in our pipelines, but when we want to ask a quick question, and rather than open those decision support tools, many of us go straight to Decipher. And one of the strongest points of Decipher is that it does not need your variant to be into Decipher for you to interrogate the data. So you can put your variant by using the transcript and the CDOT nomenclature, and Decipher will give you information on your variant. It will link out to different data sets. So if we go to the next slide, you can see an example of what does it look like when you conduct a gene search rather than a variant search. So in this case, we have the ATRX gene, and you can see there on the landing page of Decipher the links to so many data sets that are really useful, like OMIM and gene reviews, and you can link out directly to some of the ClinGen resources, see some metrics data on your gene, and etc. So incredibly useful. Next slide, please. And now I just wanted to spend some time looking at this feature of Decipher that many of you might know, but really the people I talk to time and time again say that this is one of the most useful things when you are trying to evaluate your variant, because we often are worried about the characteristics of the specific variant, but to really understand the impact of a variant, we need to understand the context, the wider biological context. And this protein view from Decipher really offers that at a very high level. So if you look at some of the labels that are on the left-hand side of the slide, you can see the labels to the tracks there. So you can see the exon track, where you can see if you're looking at the nonsense variant, for example, does it land in the last exon of the gene, or is it predicted to be target to nonsense-mediated decay? You can see a NOMAD track there, where you can very briefly see if you are landing in a region of the gene that has a lot of NOMAD variation or not. I highlighted the box where you have the ClinVar variant. So that information is all in Decipher. And for those of us that like to see things visually and graphically, this is really powerful to assess your variant. So I have one more slide where I have my conclusions. So can I have that slide, please? And really, very briefly, I hope I convinced you that we are now at the stage where many of our data sets are doing multiple functions, are linking out to other data sets. And for us, clinical scientists, that's incredibly useful, because it reduces the time that we spend opening different links and typing different web addresses. And really, all of those data sets are really helpful to contextualize a lot of the information that we want to make sense of from a biological point of view. So thank you very much. Sandy, over to you.
Thank you, Julia. Wow, what a great, vast amount of knowledge there, and such a great, useful resource. So thank you for taking us through that step by step. Really important. Okay, if we move on now, please, Emma. It's a great pleasure to now introduce our next speaker, so Swati Toma, who has a PhD in biomedical sciences from the University of Carolina in America, and then went on to complete a postdoctoral fellowship at the National University of Singapore, focusing on next generation sequencing, and then obviously is very, very well placed to be able to understand the challenges of interpreting the vast amount of genomic data. So she currently is the product manager at Sophia Genetics and is going to talk to us today with a colleague, Rolf Funt, about enhancing variant interpretation. So Swati, over to you, please.
Manager at Sophia Genetics, and I'll be talking to you about enhancing variant interpretation using Alamode Visual Plus. Alamode Visual Plus simplifies and streamlines the analysis, and it's basically a comprehensive genomic browser which helps increase productivity in a lab with reduced turnaround time. Now, this tool can be accessed both as an add-on module on top of Sophia DDM platform, as well as a separate standalone product. Now, the key features of Alamode Visual Plus are, first of all, splicing prediction scores. We have in the tool 4 in silico predictors for splicing prediction, both at donor and acceptor sites, as well as predictions for branch points. In addition, we also have in silico tools which predict the binding sites of exonic splicing enhancer proteins. Having said that, we have comprehensive variant annotations starting from GBS nomenclature, predictions for SNV indels across different population databases and in silico predictors, ACMG point-based classification, and also mutation-specific search tools, for example, Mastermind and Google Scholar. Now, this tool can take in several different file formats from BAM, CRAM, VCF, etc. And it allows the user to access and actually manage their data by creating, importing several different types of files. They can create user profiles and share those settings and the database across a specific user group in the institute. Lastly, we can also create custom report in Alamode Visual Plus, and the user can choose what to show or not show in the report, and they can download it as a PDF or HTML. Now let's take a deep dive into some of the features, starting first with splicing scores, as well as the resolution of splicing mechanism that is offered in Alamode Visual Plus. Now, on the right, you're seeing in silico prediction of deleteriousness and branch points, which is predicted by MaxNScan, splice site finder like NNSplice and gene splicer tools. And we also have the prediction for known constitutive signals, as well as the high confidence branch points. And then lastly, we have the exonic splicing enhancer binding site predictions. We also have a third-party tool integrated into Alamode Visual Plus, which is called XSkip, and it predicts the exon skipping probability of the given variant. Moving on, we have literature-specific search, a mechanism in the tool where you can use Mastermind Genomenon Track, and you can visualize how many variants are specifically present for your, how many papers are present for your publication. And then you can investigate further by clicking on the Mastermind link and curating those variants through the Mastermind Premium access. Now you can also visualize genomic context or CNVs and complex structural variants in the tool. We offer DBVAR, Decipher, Nomad, Orphanet, and MedGen as datasets where you can correlate and visualize your genomic context for a given structural variant or a copy number variant against these databases. And Decipher and DBVAR also enable the user to filter the database based on pathogenicity so that you can compare previously known pathogenic variants and visualize them. Here is a visual which shows that you can upload several different BAM and Sanger files as multiple tracks for a given case, and you can do a validation of both NGS as well as Sanger in the same view. Lastly, we also have account-specific database management, which is offered through profile settings and private annotation databases, as well as local variant databases, which are specific to different file formats that can be taken in. Now with this, I would be going into a demo case of Alamut Visual Plus and how it actually unraveled a structural variant, an allo insertion, as well as a deletion in BRCA2 gene. And I'm going to go about visualizing both different types of variants in a given case. So with this, I would like to show you here, this is the BRCA2 gene, which is visualized in Alamut Visual Plus. And I can actually go into the tool. So with here, we are looking at a BRCA2 insertion. So I created this variant by using the option of variant and new variant, and I added the insertion under the BRCA2 gene. And right now we are looking at the annotation across several different databases, including ACMG, as well as checking the visualization in the splicing tab. If I do click on the splicing tab, although it's not a splicing variant, I can visualize how the insertion has taken place. I can minimize, and this provides the user a visual context of how the insertion took place in the given exon. Now, if I hover back over to the annotation, I can also see that there are some literature which are present through the mastermind. And also I can display this in the genomic view to see the exact location where the insertion happened. Now we have several different tracks here. We have ClinVar, Genomenon Mastermind, Cosmic, Unipro, Structural Variant, and so on. And if I click on the structural variant gear sign, I can actually filter both Decipher and dbvar based on pathogenicity. And I can choose to see only the pathogenic variants here. And if I do that, now if I hover over here and I know this one is an insertion, I can right click and see what different types of variants are present in Decipher, dbvar, and Nomad. And based on the position, I can choose to open either of those patient-related variants and correlate with my variant of interest. Now, if I scroll further down, I do have this track of repeat masker, which is giving me information on some of the different types of repeats that are pulled through the repeat masker. And in this case, because we knew this was an allo insertion, it actually also overlaps with allo insertion, which is a sign track, in addition to a LTR and other simple repeat tracks. So this gives me a very good visual overview of what that region looks like and how it corresponds to several different databases. Now with this, I want to go into the second variant that the same case had, which was a deletion, and it was an in-frame deletion. And again, I had created the variant by using the variant option of new variant. And if I visualize this, in the genomic view, again, I can pinpoint to the exact location of the deletion, and I can hover over different tracks, whether it's Decipher or dbvar, and right click and see what different types of deletion have been reported in the same region. With this, if I go back to the annotation panel and look into the ACMG, I can choose to use my user-defined ACMG rules. If I'm not happy with the selection here and I want to apply ClinGen guidelines for whichever gene, I can choose this based on the metrics or evidence that I would have. And then this gets highlighted. And in the next version that will be released soon, you can also save the strengths in the annotation itself. So with this, I'm going to go back to our presentation. Now, summarizing today's talk, I would like to highlight top 3 key advantages of Alamut Visual Plus. Firstly, it helps in establishing efficient lab workflows. And whether it's related to Sophia DDM platform as an add-on module, or whether it integrates in your in-house workflows, it provides that end-to-end variant assessment that a geneticist or a variant curation scientist seeks. And it also provides comprehensive variant annotation across more than 55 different databases. And it actually enhances the variant visualization across all these different databases. In the end, it saves time and improves productivity with all tertiary analysis tools presented in one user-friendly format. Now, with this, I would like to have just one slide dedicated to what's next in Alamut Visual Plus. And I'm happy to announce that in the next release we're going to have later this year, we will have shared database on cloud. We will have a new database of MitoMap, new splicing predictor, and also alpha missense. If you want to know more about these features, please feel free to reach out and we would be happy to talk more about these. With this, thank you, and I hand over to Rolf, who will talk about their clinical workflows at Radboud University Medical Center. Thank you. So, hi, Rolf. It's a pleasure to meet you. And would you like to please introduce yourself and tell us a bit about your role in your current institute and what do you do in terms of clinical NGS?
Okay,
Thank you.
yeah, so my name is Rolf Punt and I'm a clinical laboratory geneticist at the Department of Human Genetics of the Radboud University Medical Center in Nijmegen, the Netherlands. I work in the division of genome diagnostics and we perform diagnostic testing for a large number of rare diseases, but also prenatal and postnatal cytogenetics, for instance. And I work in a team that focuses on patients with intellectual disability and patients with multiple congenital anomalies. So yeah, we are performing genetic testing for those patients. And I think in the last, I think probably over 20 years ago, there was a big shift from regular cytogenetics to array diagnostics. So that was the moment when I started working in the genetic field. And I think, well, probably 10 years ago, there was a big move from regular sequencing towards next generation sequencing. So, and that's the main technique that's now being used for both sequence variants, but also for copy number variants.
Thanks, Rolf. And sorry, and you mentioned NGS applications. So could you just elaborate on what all types of NGS applications are you currently routinely using?
Yeah, I think we, as an institute, made the choice when NGS was implemented in the diagnostic setting, not to let's say to make focused gene panels only, but we started with whole exome sequencing from the beginning, and then let's say use gene panel information to perform targeted analysis. And that was very successful in the sense that it replaced a lot of other techniques. Well, we started with a few gene panels in the beginning, but now that has grown to many more disease indications. And I think we are trying to direct the flow of all genetic testing as much as possible towards this exome sequencing. So even for, let's say, if we find a variant in a patient and the parents or family members are sent in for that specific variant, in any case, we will still perform exome sequencing because it's just the regular flow.
Right.
And I think 1.5 years ago, we also started implementing whole genome sequencing. But that's for a limited number of diseases.
And these are done in addition to the exome sequencing, or is it?
Well, in addition, let's say, as for instance, for intellectual disability, we implemented a whole genome sequencing. So that has replaced the whole exome sequencing testing for those patients.
Thank you, Rolf. And now I would like to understand, because we know that you're using Alamut Visual Plus in your variant interpretation workflow. So I was wondering if you could elaborate what are the biggest benefits of Alamut Visual Plus that help you overcome your current challenges in variant interpretation?
Right, yeah. So I think we're already used to use Alamut for targeted sequencing. So for, let's say, the traditional Sanger sequencing analysis. So we were very used to see a variant in the context of the genetic information and of the genomic context. And well, we've continued to do that also for the next generation sequencing applications, because let's say the visualization of the variant in the context of the gene and also all the information that you are provided with, that helps a lot in making the interpretation. So if you're just looking at a table with the variant, that just gives you a limited number of information sources. And of course, you can also look in various public databases on the internet, but especially the visualization of the variant together with the data sources, that helps a lot. And of course, also in Alamut, you're also helped with the nomenclature of the variant, which you can, of course, adjust or check if you agree with the nomenclature, but that you can use it to feed your own local databases or your clinical reports. Well, also it helps you with the classification in the sense that in the latest version, also the ACMG classification is implemented. It's not perfect, but it's a start. It helps you with to have everything in one place.
Right. Thank you, Rolf. And I was wondering if you could speak a little bit about how does Alamut Visual Plus streamline your lab workflows and supports in clinical research efforts, and you could provide some context also here.
Yeah, so I think like I mentioned before, it provides more or less the link between the sequencing data that is provided by the sequencing lab and the bioinformaticians, and let's say the databases that you have available. And it's the link between generating the sequencing data and doing the interpretation of the data within the context of the gene. So that's the main advantage. It's more or less like I mentioned previously. It's to you. It's for me. It's actually the missing link between the data and let's say all the annotations that are available. But then to really make the interpretation and to see where the variant is located and get all the data together in one screen. That helps a lot.
Thank you, and thank you for saying that. Like, it's that missing piece in your puzzle, right? Then it completes the picture and acts as a bridge between, let's say, just the file and the endpoint that you're seeking.
Yeah. So that's also because we get a lot of questions also from the bioinformaticians from our department. Why are you using it? Because you have all this data that we provide you with all the columns, but we want to see the context of the variant. But that's
Thank you, Rolf. That was super helpful. And now, if you could give an example of any challenging or a complicated case that you were able to solve using Alamut Visual Plus, we would like to hear more about that.
Yeah, so there's one specific case that pops into my mind that let's say there was a patient with a severe phenotype for which we performed exome sequencing analysis, and we sent out a negative report. And then I think a few weeks later, the referring clinician called and said, well, we had a meeting together with different disciplines, and we looked at the patient again. And we think it might be that this patient could have a mutation in a specific gene, the UFM1 gene. Could you please take a look at that gene to see if there might be a mutation that you've missed? So I went back to the annotated data table of the NGS data, and I searched for the gene, and there was no variant present in that gene. So I said to her, no, I'm sorry. No variant present in the gene that you suspect. Okay, there was that, and it turned out that she sent all the details of that patient to a neurologist in Amsterdam, who is let's say a leading figure in the field. She also took a look at the data and at the patient, and she said, well, I've looked at the patient and I'm convinced that she must have a variant in this gene, and I think based on her name, I think she might be from a Roma ethnicity, and there's one founder mutation in that gene that's located far 5' of the gene itself at minus 273, I think. She has to have that mutation. So we went back to the data, first to the data table. I couldn't see it. I went back to then I opened all the data in Alamut, and then I think in just one glance, I could see, oh my God, there's this mutation in this gene located far in front of the gene, but still she had that specific variant in a homozygous state. Both parents were carrier. And it turned out that because of the annotation that's being used, I think not only by our institute, there was also a non-coding RNA gene at that position.
I see.
And the variant was present in the sequencing data, but it was not annotated to the gene itself. So if you could search for the gene, but you will never find the variant. Whereas if you look at the data, you could see there's a ClinVar variant, there's a human HEMD mutation at that location, and exactly in that position there was also the mutation in that patient. So that helped me, and also if people call to check a specific gene.
Yeah.
Not only looking at the data in the annotated data table, but also visualize it. And you could of course also use IGV for that Well, if you look in the IGV for that specific patient, you would never correlate that mutation to the gene because it's far 5 prime.
And that's a quite interesting case, Rolf, because in one hand, it elaborates the challenge of variant annotation pipeline, which can be, user specific or workflow specific. And then there is another, let's say, the missing links where you bring in extra pieces of information and then you sort of seek the complete picture. And it's perfect the way you could resolve the case. Thank you so much. And finally, if you could please walk us through your typical variant interpretation workflow, and you can share your screen at this point.
Okay, so we are using a in-house developed data analysis software package, which I show you here. And this actually is the data of the patient that I just mentioned with the UFM1 variant that we missed in the first analysis. So all the data is available here. You can see here, in this case, almost 140,000 variants in this whole exome data analysis. And we can use a number of different filter combinations to perform filtering on the data set itself. And then after filtering, you're left with a number of variants that you need to look in detail. And we go to a number of consecutive steps in our protocol. Let's say the filter combinations, the first one is, of course, if you have trio data available, we would like to look at de novo variants that are present in the data. So you're left with a small number of variants, and those variants are looked in detail, also using data sources and, for instance, also Alamut, but also variants that are very rare and already known in a disease database like class 4 or 5 in ClinVar or in our in-house database, but also rare variants that are known to cause a truncation, frame shifts, nonsense variants. And then, of course, we are also looking at rare and recessive. So we have a number of different filter steps. And once we have a number of variants that are left after filtering, we can look in the data analysis software and use our right mouse button and go to any data source that is available. And for instance, also the Alamut software. I would like to show you the patient with the difficult variant in the UFM1 gene. So this is the data from that specific patient. And if someone would ask for the analysis of a specific gene, we can just type in the name of that specific gene and look for any variants. And you can see that there are a number of variants present in this gene in the patient data, but these are all intronic and a silent variant with high GNOMED frequencies. So these variants are of no interest. But we could also go to Alamut and look at the patient data. Okay, so here you can see the UFM1 gene, and we've also loaded the sequencing data of the patient and both parents. In this case, also in Alamut, you can see here the BAM files of the patient and both parental samples down below. And the nice thing is that you can, because all the data is available in the Alamut software, you can see here, for instance, the ClinVar variants. And if you would look at the ClinVar variants, you can see that the screen is now from before the first non-coding exon and after 3' UTR, that there are also variants present in front of the 5' UTR.
Right.
And actually, there's actually one deletion insertion way in front of the 5' UTR. And this is exactly the variant that's present in this data. So if we zoom in, you see where it's at, minus 270 something. So this is the ClinVar variant of interest. This is the founder variant that's found in the Roma population. And if you now go down to the BAM file of the patient, you can see this is the HGMD. We have a license for the human gene mutation database track as well. So we have that loaded in Alamut as well as our local information track. And you can see here the 3 base pair deletion in the patient in a homozygous state and both parental samples down below, and they have this same variant heterozygously. So because you now can see the entire context of the gene, this variant is now far more easily visible and it's not present in the data table, I can show you and the annotation of our data. Go to the. So here's an Excel view of the exome data that's presented in the analysis software that we use. And here you can see the name of the UFM1 gene. So these green variants are annotated to the UFM1 gene. And this variant in orange, that's the actual variant of interest. It's also in the human gene mutation database, but this is actually not annotated to UFM1 because it's before the 5' UTR, and it's actually annotated to a non-coding gene.
Yeah.
And that's the reason why we're not finding it if you're looking for this gene, because the variant is not annotated to this specific gene itself. But if you look at the data in the context of the entire gene in Alamut, then you can see that there are described variants, pathogenic variants, also outside of the coding region.
Thank you, Rolf. This is super interesting how you could actually narrow down onto your variant of interest using multiple sources and then eventually visualizing the genomic context. And did you also do the variant classification, the ACMG, or the variant annotation panel that we have in Alamut Visual Plus? Do you use that as well in addition to the visualization?
Well, we use it as a as a help, more or less, but we are still one version behind, right?
I see.
And
Yes.
there was a bug in that version. So if I make this deletion.
Okay, so you make the deletion. Yeah.
Yeah. So I think I'm not sure if you use the ACMG classifications. This is also for this specific variant. It's difficult because it's intronic.
Correct.
But
yes.
in this case, because it's a founder variant that has
Right.
already been described, then you
Right.
will be able to classify it as such.
Interesting. Yeah. Thank you so much.
When asked which patient, what element made a difference in the diagnostic setting, I had to think about this patient. So I'm very glad that I could show you the details of that analysis and of that specific variant. And just I think to keep in mind for myself, but also for other people doing analysis, that the annotation of the variant might not always be correct. And if you could just look at it in the context of the gene, that might help a lot.
Thank you. Thank you so much, Rolf. And we are glad to have you here explain your routine variant interpretation workflow in a clinical NGS setting. And it's always a pleasure to understand how Alamut Visual Plus is being used in different settings and how it can address their challenges also. So thank you so much.
Thank you for having me.
Thank you very much indeed. That was incredibly insightful. So let's now move into the live Q&A session if we can. We've had quite a few questions being submitted on the different sites. So first of all, I'm going to come to Julia. Question on you about use of ClinVar. So if you have additional information to reclassify a variant that conflicts with what ClinVar is saying, so the submissions are already there, can you and should you share that information with ClinVar?
Oh, yes, absolutely. Yes, you should share this information. And there are different ways of doing that. I guess there's one way where many laboratories are already submitting all of their variants to ClinVar, so that is a way. But the other way is to be part of one of the ClinGen expert groups. So if you have a lot of expertise in a gene or in a condition, you can go to ClinGen and apply to become part of one of those expert groups that then feeds that information into ClinVar. I think that is really fundamental because as we gain new knowledge, it's important that we collaborate and share that knowledge. So yes, definitely.
Great, thank you, Julia. And I think you're right. The more we can share and learn, we all benefit, particularly in the future. Thank you. Svati, that was fantastic to just really see Alamut being used in the real world. And I know we don't endorse products here, but I think we know that many laboratories do use that system. We've got one question coming up, and I think we all are struggling with informatic information governance and GDPR compliance, etc. So we're saying GDPR and information governance issues may inhibit the use of a cloud-based variant database for anything other than aggregated patient data. And that's about identifying an individual if you're sharing phenotype as well as genomic data. So will AVP version 2 be available with a locally held database option?
Yeah, thank you so much for bringing that question, Tony, and thank you, Sandy, for elaborating on it. So when it comes to GDPR and HIPAA compliance, our data processing and storage is compliant with that one, and then we have some additional ISO certifications just to enable that the data can be, processed and saved and just accessed through cloud. But we are also going to work very closely with institutes who will be transitioning and migrating their databases from local to the cloud. So we will address the nuances, let's say, to get that clearance from the InfoSec department of the respective institutes. So in terms of the local option, there will be a period of transition where we will address all these issues and all the teething issues can be clarified, and then we migrate.
Great, thank you. I think that would be welcomed by many, many laboratories in many, many different countries as well. Thank you. One for the EQA team. Do you expect laboratories to state the variant decision tools that they've used on their reports, or is that not appropriate or not required? What's your thoughts? I don't know who wants to go first. Dave?
I'm happy to answer that.
Or
Yeah, so
Barra?
it's not a requirement for them to do that. Often people keep this information in-house because really it can make a report really long, and you want the clinically relevant information just there for the reader of the report. So often it's not included.
Great, thank you, Farah. Becky, I'm going to ask you as well the one about variant interpretation or classification, because you've been heavily involved in providing the GINI, the individual classification variants. When it comes to the use of different criteria, say ACMG criteria in particular, is it variable, or everybody pretty much know what they're doing now and it's pretty easy stuff?
I think it's very variable, and I think particularly, obviously, I've been involved with the BRCA and HRR gene variant classification assessments, and these are offered globally, and we have a huge number of participants in each run, like 500, and still the variety of guidelines and things that people are using is still very wide. And so consequently, the variation that you get in evidence being used and even the classification obtained can be very variable for a single variant.
Thank you, Becky. So that's delivered, GINI's delivered to individuals. Dave, you run the EQA's laboratory-based ones for the hereditary breast and ovarian cancer and Lynch syndrome, etc. Do you find the same, or at a laboratory level, is it more standardized?
It can be, say, if you're looking within a specific region or a country, or there tends to be a bit more agreement. But I think there's still a huge variety across all the labs that participate in our EQAs. And I think some of it stems from the guidelines. There's been all these additions made to the guidelines over the years, and we're all kind of waiting for the new ACMG guidelines. But I think different people have chosen to adopt different guidelines. Different countries have gone, down their own routes with some, advisory things they've got. I think it's kind of inevitable. You'd like to think that the overall classification that people get to at the end would be similar. I think sometimes we get a lot more variation around the uncertain significance, likely pathogenic, likely benign. I think there's more variation there. Once barn door, pathogenic, benign, we don't see quite as much trouble with. But yeah, huge amount of variation.
Yeah, thanks, Dave.
And can I just add to that, Sandy?
Yeah.
Sorry, that sometimes labs have cases in-house which can, give help change the classification for a variant as well. So that's all taken into consideration because they submit the evidence as well. So assessors all take this in. So there could be a reason why one lab submits a class 4 and one the same variant class 5. So everything's taken into consideration when assessors look at this.
Great, thank you. And that actually leads into the last question I'm going to ask both to Julia and Swati. So Julia, first of all, so same question to both of you. So the ClinGen expert groups don't have access to all the information available for a variant, and that comes back to Farah and Dave's points about, and Becky's point about labs maybe have their own database or specialist knowledge in particular genes. So would you recommend that rather than taking whichever classification or information comes out from a decision support tool to be gospel, or should you really use your own knowledge as well? So Julia, you first, and then Swati for your thoughts.
It's an easy one, Sandy. You always have to use your scientific knowledge. I tend to tell trainees that this is not mathematics, this is biology. So it's not, you can't put things in a calculator and get the right answer. Everything coupled with scientific judgment. We see things that are rare and unusual. So we need to take all of that into account and our own expertise that, and when we don't know something, then we should reach out to our colleagues and those who do know and have that expertise to make sure that we don't get the wrong answer.
Great, thank you, Julia. Swati, what's your thoughts?
Yes, absolutely. Thank you, Julia, for answering that question, because I completely agree with what you just said, because the discretion of the variant scientist and the context of the disease, the context of ethnicity, and the phenotype spectrum, there's so many angles to the interpreting a very single variant, and that's what makes it challenging, because there's no set rule book to address these variant classifications. So they are definitely guidelines. They're not gospels, and the user has to take it with a grain of salt. But the good thing is they do have the evidence in front of them, and then they can have all the tools to make that final decision based on all the review, because ultimately every variant curation does go through a committee where they sit with the variant curation scientist, the bioinformatician, the clinician. Okay, this is what we have. Are we all in agreement? So yeah, it has to be, that process has to be followed.
Great, thank you. So I think the take-home message is we're all still learning, and will be for quite a while just now, but I think we have a lot of decision support tools out there that really can help make it easier and try and pave the way forward for standardization. So thank you everyone very much indeed. What a fantastic session, great amount of information and education there, and quite a fruitful discussion as well. So thank you very much indeed. Emma, do we have a slide for the next Focus On webinar? Lovely. So we very much perform these webinars in response to participant demand and sort of asking you what you would like us to discuss and present, and get some key experts to come and speak to you. So please do let us know if there's anything you would like us to focus on going forward. The webinar will be available on the GenQA YouTube channel in about a month's time. It takes a month for it to go on there, but you'll automatically be sent a participation certificate for joining us today. Also, just to keep up to speed with what's happening, there's always new pilots on the go, workshops, webinars, and if you want to, you can sign up to our newsletter to get that information into your inbox. And our next Focus On topic will be on implementing a national fetal sequencing services, benefits, the challenges, and hopefully the solutions to all those problems, by Professor Dame Lynn Chitty from Great Ormond Street Hospital in North Thames Genomic Laboratory Hub in London in the UK, and that's on the 12th of November. So please sign up if that's something of interest. And thank you again to our panel and our EQA providers today. Thank you.
Thank you. Bye-bye.